Section 15.3, found on pages 296 to 297. We are going to learn about mapping the distance between genes using recombination data. If you remember from earlier in the section, recombinant offspring, or recombinants for short, are offspring with combos of traits that differ from those found in either parent. For an example from Mendel's pea plant experiments, we could cross a parent plant with genotype big Y, big R, little r, that bears a yellow round seed, and cross it with a second parent plant with genotype little y, little r, that bears a green wrinkled seed. Parent 1 could provide gametes big Y, big R, little r, big Y, little r, and little y, big R. The only gametes that parent 2, the green wrinkled homozygous recessive parent, could provide are little y, little r. These parents could produce offspring with big Y, big R, little r, yellow round seed-bearing plants, little y, little r, green wrinkled seed-bearing plants, big Y, little r, yellow wrinkled seed-bearing plants, and little y, big R, green round seed-bearing plants. The yellow round and green wrinkled seed-bearing plants would be known as the parental type offspring because they show combinations of traits identical to those of at least one of the parents. The yellow wrinkled and green round seed-bearing offspring would be what we're concerned with, the recombinant offspring, because they show combinations of traits that differ from either of the parents. Recombinant offspring result from crossing over during prophase 1. Using this information, you are ready to learn all about mapping genetic loci on a chromosome.
Alfred Sturtevant was a student of Thomas Hunt Morgan, who used genetic maps that were produced by the discovery of linked genes and the recombination theory. He believed that the recombination frequency was determined by the distance that the genes were located on a chromosome. So if this was your whole chromosome, these individual genes have a different distance between each other. He thought that crossing over occurred along all points of the chromosomes, and he predicted that the distance between two genes increased the probability of the crossing over occurring. So, for example, these two genes would be less likely to cross over because they are closer together, but these two genes would have a higher likelihood of crossing over because their distance is further apart.
One type of genetic mapping is called a linkage map. These are based on things called recombination frequencies. A man by the name of Sturtevant conducted an experiment to prove these. In his experiment, he concluded that a linkage map is a technique that is based on assumptions that the possibility of a crossing over of two gene loci is proportional to the distance separating the loci. The distances are expressed by map units. One map unit is equal to a 1% recombination frequency. And for an example, if you had a chromosome with your gene loci being here and here, and the distance was, say, 6 map units, that would equal a 6% recombination frequency. These recombination frequencies are used to make a linkage map for one specific chromosome. Genes are then arranged on the chromosome in the order that best fits the data. Sturtevant concluded that he observed the recombination frequencies between the 3 Drosophila genes, which are little b for body color, CN for cinnabar or eye color, and VG for wing size. Due to the recombination frequency of 9% between B and CN, and the 9.5% recombination frequency between CN and VG, Sturtevant concluded that CN would be positioned halfway between B and VG. So therefore, he concluded that the crossovers between CN and B and CN and VG are half as frequent as the crossovers between B and VG. So this map does locate that CN is halfway placed between B and VG. Although this example is not very difficult. Don't worry, there are more difficult ones. Sometimes there are genes on a chromosome that are so far apart that the possibility of a crossover is unlikely. Now some of you may be wondering what the max recombination frequency is. And the max frequency of a crossover for gene loci is 50%. Now with that, the physical connections between the genes on the same chromosome are not reflected. To the results of a genetic cross. Being physically connected is just as good as if they were genetically unlinked. Being genetically linked is where alleles of genes would assort themselves independently as if they were on different chromosomes. As a matter of fact, when Gregor Mendel was studying his pea plants, he realized that there were two traits that showed up on the same chromosome. However, though, the distance between them was so great that the linkage was not observed on a genetic cross. Therefore, the two genes behaved as if they were on different chromosomes. Genes that are located far apart on a chromosome are mapped by adding the recombination frequencies from crossing over pairs of genes lying between the two distant genes.
Using recombination data, Sturdivant and his colleagues were able to map numerous Drosophila genes in linear arrays. They observed that the genes clustered into 4 groups of linkage genes, known as linkage groups. By using microscopes, we know that these fruit flies have 4 pairs of chromosomes, 3 pairs of autosomes, and 1 pair of sex chromosomes. This means that linkage maps provide further support for the theory that genes are located on chromosomes. Each chromosome in the 4 pairs has a linear array of genes specific to that chromosome, and each individual gene has its own specific locus on that chromosome. For example, the gene for whether the fly has a black body or a gray body could be located here. Linkage maps can be drawn to show the estimated loci of the gene with respect to the other genes. The numerals above each gene locus signify the number of map units between it and the gene on the far left, the gene for arista length. On this map, we can see that the gene for body color is 48.5 map units away from the gene for arista length, whereas the gene for eye color is 104.5 map units away. It must be noted, however, that linkage maps give only an approximated picture of a chromosome because it is based only on recombination frequencies. The frequency of crossing over is not actually uniform over the length of a chromosome, meaning that map units do not correspond to actual units of distance, such as nanometers. While a linkage map is useful in portraying the order of genes along a chromosome, it cannot accurately portray the precise location of the genes. Instead, geneticists construct cytogenic maps of chromosomes, which locate genes with respect to unique features in a chromosome, like stained bands similar to the one seen in this image, which can be viewed with a microscope. If you were to compare a cytogenetic map with a linkage map, you would see that once again, the linear order of the genes is identical in both, but the spacing between genes is not. This is an example of a cytogenetic map. The centromere separates the chromosome into 2 arms, the short arm and the long arm, designated P and Q respectively. The arms are separated into regions, as seen in this region number 3, and then further separated into bands. The bands represent the alternating light and dark portions, such as with this black and gray area next to this white area. These bands can be further separated into subbands, bands that alternate color within a light or dark portion, such as the difference between this black subband and this gray subband. By giving each of these characteristics a code, we can map out the exact location of a gene, as seen with the CFTR gene on this organism's chromosome. We can trace the CFTR gene to a specific chromosomal location, given by this code, 7q3.1.2. By using this code, we can see that it's located on chromosome 7, on the long arm Q, within this region 3, on the first band, and the second subband. That way, we can narrow it down to this precise location on the chromosome. Cytogenetic maps provide far more accurate data for the specific location of a gene than linkage maps.
To sum up, Alfred H. Sturtevant came up with the method of using genetic maps from the discovery of linked genes and recombination. Sturtevant's idea was that the recombination frequency was based on the distance between genes on a chromosome, with his assumption that the chances of crossing over equal at all points along the chromosome. He was able to predict that the farther apart two genes are, the higher the probability that a crossover will occur between them, and therefore the higher the recombination frequency. For example, these two genes on this chromosome have a higher chance of being separated than these two genes on this chromosome, due to the fact that there is more distance between them. In other words, there are more points along that chromosome between those two genes where crossing over can occur, thus causing the chance of those genes being separated to be greater. Let's look at a quick example to help understand this concept better. If the recombination frequency between gene A and gene B is 15%, and the recombination frequency between gene A and gene C is 10%, what are some possible linear orders of these three genes along a chromosome? If gene A and gene C's recombination frequency is 10%, we know that gene B cannot be between those two genes. Therefore, gene B must either be before gene A or after gene C. Sturtevant was then able to use this information he gathered to map the genes on genetic maps called linkage maps. These genetic maps only portray the order of the genes and not the actual distance between the genes. A more accurate depiction of chromosomes and their gene loci locations comes from cytogenetic maps. Cytogenetic maps use chromosomal features to map out the exact locations of the genes along the chromosome.